Let me now welcome Professor Graham Ball, who is a professor of bioinformatics at the Nottingham Trent University in UK. He works in the interface of how omics technology and the big data set could be utilized and analyzed using different bioinformatic tools, and also make sense of them using machine learning, artificial intelligence based, current buzzword which you are hearing, how to make use of them to make good prediction of the panel of the biomarkers. So we do a lot of joint work on different cancer project and different biological projects, where we are really privileged to have his inputs and his various skill which he is utilizing to do the bioinformatic data analysis. Just to connect the dots, what we are going to do now, Professor Graham is going to talk to you about how can you access some of the data sets at the genome-wide scale level. So I talked to you about the significance of the central dogma of life, how DNA, RNA, and proteins are important. Now, if you really want to understand a life science system, any given organism like human or plant or any given organism of your interest, you want to know the bigger picture, what is happening in all the genes of that given organism. So you want to, if you just study 10 genes specific for heart or 10 genes specific for lung, you can get some information, but it's specific to that particular organelle or that particular organ. But now if you are looking at the entire human body, entire human system, you should be looking at much bigger picture, and that's where it's a good idea to look at genome-wide scale screening. And that's where omics technologies like microarrays are important. Now, while we do a lot of experiments, wet lab experiments in my lab at IIT Bombay, and we generate a lot of data and we also provide those data to the community back. Likewise, there are many groups in the whole world who are generating lot of data, omics type of data set. And it is now a good custom for people to make that data publicly available. So you have a lot of public data repositories from where you can go and access these data set. So again, if you are specialized more into data analytics, big data sciences, you need not to rely on the biologist to generate the data or to provide you data. You can always go back and try to use these resources and generate data, download the data from these places. All you need to know that how to analyze this data using different type of open access software, or you can write your own programming and do those analysis. So Professor Graham is going to take you from those modules of how to take the public data repositories, places like ArrayExpress, how to access those, generate those data set, and then how to analyze those and make meaningful sense, that we will talk as we go along in the course. Professor Graham, sorry for a very detailed introduction and the context, but now I think the stage is all yours to take forward.
Thank you, Sanjeev, and thank you for the opportunity to present to this group. I'm just going to share my screen. So. Can you see my screen okay?
Yeah. You're audible and screen is seen.
Okay, so good afternoon, everybody. I'm speaking to you from a quite dark and cold morning in the UK at the minute, but anyway, I'll move on because that is another issue. So I'm going to talk to you a bit about public data repositories, and this is a really important issue because it means that by accessing these data sources, it means anybody can do research into understanding the nature of biological diseases, biological systems. And we have moved considerably since the sequencing of the human genome in 2003. We've moved massively on. Before that, people would look maybe at 5 molecules max and try and understand the whole biological system by looking just at 5 molecules. At that point, it was very much a sort of hypothesis-driven approach, whereas since the sequencing of the human genome, we're now able to measure the message, or measure the nature of every gene, the message coming from that gene, and how that message relates to the proteome and ultimately relates to biology. So we've moved from a hypothesis-driven approach really to a data-driven approach. And there's great opportunities here, because if we can measure every molecule in the system, then we can get a more complete understanding of the biology of that system. So one way to achieve this is by people sharing their data. And this is done through various public data repositories which are hosted on the internet. There are also often private data repositories where you can join, and you may be able to join, for example, by just signing up to a form or agreeing to some terms and conditions. And things like in the UK, we have the UK Biobank. That's a classic example where they have tracked the data for around 40,000 individuals who are ages of 50 or more, and they track all their medical records, and they have also data on their health, lifestyle, and molecular data as well. So there are actually requirements to share data. So these are often written into the terms and conditions of, for example, grants and grant applications. So if something is funded by public money, there is often a requirement to share that data. There's also a requirement to share data as part of a publication, for example. So if you have a high impact journal, or most journals in fact now, will require you to share your data that you came to the conclusions in the paper on a public repository. And of course, there's the main benefit, which is the benefit of the data to mankind. So the altruistic use of the data to greater understand different states. So in order to share data, we have to use a standard. And that standard is, or started out as the Miami standard, which is minimum amount of information about microarray experiment. And so basically this sets out guidelines for what has to be included in the repository for the data sharing. Now, as the next generation sequencing has come about more and become more available, then there's also the MinSeq standard, which is a minimum amount of information about, next generation sequencing. And this is really defining when we share our data, what we should include. So the Miami standard requires that we share our raw data. So if it's an expression array, then that means we share the images of the array, the cell files. Or if it's sequencing data, then we have to share the FASTQ files, which are the raw files with the sequence information, not necessarily aligned or shared in a form that's aligned to a genome, but in its raw form. There's also a requirement to share the processed and normalized data. So frequently we normalize this sort of data based upon the expression level of housekeeping genes, or the account of the number of genes copies in that system. So this is important, because you can imagine from a sample we might get different amounts of RNA or protein out, so we have to standardize that. We normalize to that by looking at either the amount of material or the expression level, the amount of message coming from a few standard genes, which we call housekeeping genes. We also in the standard have to state the essential sample information. So for example, what was the tissue that was profiled, or maybe blood, tissue, other body samples? What was the gender of the individual who had that sample taken, and what was their age? Because these are fundamental processes which will affect the nature of the sample and how things are measured and the signature. But also we have to include, excuse me, we also have to include experimental factors. So for example, if we do a dose response study, we have to include the dose. If we look at any treatment we have made of that tissue, we have to include that. We also have to include, for example, if we're looking at particular cell types or tissue, or looking at particular disease states, and there's clinical information, we have to include that as well. Now, as you'll see when I go through some examples, there is also the sample data relationship. And basically what that does is that links the clinical information, or the information about particular samples, to the omics profile data. So what you'll often find is there's a particular code, which is the code for a given sample, and that is the code that links in a relational database, that links the sample identification through with all the clinical and recorded features through to the profile information. Now, we also need annotation, because we need to know what things have been profiled. So what gene identifiers, what genomic coordinates, the chromosomal number, maybe the sequence of the gene we're looking at. All of that needs to be recorded. So we have annotation files or array design files. And then also, for example, what methods have been used on this data? How was the DNA, RNA, protein, etc. extracted? What the method of normalization was used? What process was made to go from the sample, the way the sample was stored, right the way through to the end processed signature file. So all of this is very important, because it means that somebody who's looking at that particular study can quickly identify what's been done, what methods have been used, what the nature of the samples are, and how we can take that forward for further analysis. So often there are various types of data stored. So one of the most common ones is a transcriptomic array, but also more and more we see protein arrays, DNA-seq, RNA-seq, microRNAs, methylation arrays, and mass spectrometry data as well. So this covers the central dogma of biology. We're going all the way from DNA and right the way through to metabolome ultimately, although less metabolomic data is stored really now. So there were quite a few main repositories, and these contain quite significant amounts of data. There is no need for people to, if they want to start exploring this and start exploring molecular biology, there's no need really at the first instance to set up expensive laboratory infrastructure. A hypothesis can be tested quite easily on public data. However, we shouldn't stop in terms of our generation of data. This is really the most fundamental part. We always will need people like Professor Srivastava, who will be creating high quality data, because we are in a data-driven society. The more data we have, the more we will understand about these systems. So this covers the the main repositories. We have the gene expression Omnibus, which is the American NCBI source. We have ArrayExpress, which is the European Bioinformatics Institute ABI source. And these used to align, but now they don't, which means that actually you have samples that are stored on the EBI, which are not on the NCBI, and so on and so forth. If you have an interest in cancer, then probably one of the key repositories there is Tcga, the Cancer Genome Atlas. And that has information on around about 24 different types of cancer. And there we have transcriptomic, DNA sequence, RNA sequence, array, methylation, microRNAs. All of that data is covered, and often particular samples have been analyzed with multiple technologies. So that opens up the opportunity to compare types of cancer and to do multi-omic studies, where we look at multiple data sources and link them all together. Another big European repository is ENA, which is the European Nucleotide Archive, and that also is based at the European Bioinformatics Institute, the EBI. But this is all well and good, but I will show you and go through these repositories with you and show you how to access data from them. So often these repositories will have a front-end search. This means that you can quickly get to the data that you want and quickly access it. So you may not want to look at every single data set. You might want to sort on various parameters. So for example, you can sort on replicates, you can sort on search accuracy, you can sort on species, you can sort on the date the data was released. The data released is important because as we've moved on, technologies have advanced, and the more recent arrays and more recent technologies are much more in-depth and much more accurate than some of the early methods. We can also filter as well. So we can filter, for example, on species. We might want to just study human disease states, or the human transcriptome, genome, proteome. We also can filter on the technology. So for example, we can filter as to whether it's an array technology, sequencing technology, and we can filter on the molecule type. So for example, we can look for just RNA or just proteins. And the last thing we can do is filter on the array type. There are many different array technologies, and we might want to find studies that have just been run on the same type of array technology, which means that we can then do more accurate comparative studies between different data sets. So I'm just going to quickly end my sharing and then go into my browser, and then in order to show you the some of these repositories. So can everybody see my browser? Okay, so
yeah, it is seen. Google we can see.
Okay, perfect. So the first one I'm going to look at is Tcga. So if I click on Tcga data, that brings me up the Cancer Genome Atlas program. And so this is as part of National Cancer Institute and NIH in the US. And you can see that there's quite a lot of information. There are a range of things that are available. We can access the data. We can look at the cancer types. We can even get access to computational tools, which will allow us to look at networks and search for patterns. We can look at characterization platforms, and we can look at the timelines and milestones for this project. So the excuse me. So the first thing I really need to get is access to the TCGA data. So you can see here, this is the main portal for the TCGA. And in here, I can search for specific genes, for example, BRAF. I can search for a cancer type. I can search for a cancer type with a TCGA reference, or even a particular study. And you can see here, for example, this study has 84,000 different cancer cases in it. So it's quite comprehensive. If we look here, we can see the different cancer types as well. And each one of these reflects the number of samples. So for example, for lung cancer, there are 12,026 cases measured there. So this is quite a nice sort of splash page for this. And let's say I'm interested in breast cancer. So if I click on here, then immediately that brings me up all the studies for breast cancer. And you can see here the available files. So we have sequence data, we have expression data, we have single nucleotide variation data, we have CNV files, we have methylation, copy number variation, and methylation studies. So we have a lot of, and that's for each individual in the population. So this is a case ID. And you can see here that we have 9,115 cases. So this is a huge repository. It's quite daunting at the beginning to find our way around this, but if we start to filter down a bit, then we know that we can start to get something more useful out of it. So the filters are here on these tabs. You can see we can filter by case, we can filter by clinical data, we can filter by particular expression levels, and we can filter by mutations. So first of all, I'm going to make sure that so we're just looking at breast cancer cases. Now I'm going to look specifically at the TCGA data, which is 1,099 cases, because they've all, I know, been run in the same way. And we want the TCGA BRCA, so the TCGA breast cancer data set. Now, in this, because the many different types of cancer can form in the breast, and there might be sort of different types of breast cancer or secondary tumors, I want to just look at ductal and lobular neoplasm, so just selecting that one type. You can see also I can select on the sample types, so I just want to look at the primary tumor here. So now I've really sort of filtered down. I've still got 1,054 cases to look at, but I've got essentially a very pure population here. And you can see up here, I've got some tags that have come up. So disease type is ductal lobular neoplasms, and primary site is breast, and program name is Tcga, and project ID is Tcga, and sample type is primary tumor. So it's building up this filter term as I go through this. So I'm going to look at clinical. So, for example, I can put in gender. Male breast cancer does occur. It's not as common as female breast cancer, but I want to perhaps just stick to female breast cancer. I can look at race, and you can see that kind of reflects an American population, which is a limitation. We can look at ethnicity, and that's really just as reported as Hispanic or Latino, or not Hispanic or Latino. I can look at vital status, so which individuals were alive and which individuals died. And I can also look at risk. So, for example, I can find the individuals who succumb very quickly to their disease or who are still alive. Things like age at diagnosis are already there. Things like treatment or therapy are there. Even exposure to alcohol. So this is I'm not going to filter based on any of those. I can also look at particular genes. So I might be just wanting to pull out a certain set of genes for these. At the moment, though, I want all of the genes. And I can also look at mutations. So this allows me to filter out on different types of mutations, whether they're missense, non-coding, downstream, single base substitutions, and so on. So again, I can search on those, but I'm not going to at this point in time. So now effectively what I've done is I've put together my shopping list. So I now have the cases that I want, and what I then do is I go to View Files in Repository. And you can see here now each of these samples is either open or controlled. And we know the data type as well, and we know the data format. So it's, for example, transcriptome profiling is interesting to us. Copy number variation might be interesting. But again, I need to filter a little bit on that. So for example, I want to, in this state, I might want to just look at the transcriptome. And I might want to look at just gene expression quantification. And I want to look at RNA-seq. And I want to look at the amount of message, and that's usually done with FPKM values. So that's the count of the number of copies, if you like, or count of the number of molecules that are coming from each gene. So I'm going to select FPKM. Now, I can then select the data format, and the only option is text, but I'm going to select that, and I only want to access open files. I'll select that. So what that's now done is it's given me a series of files. You can see from here that they're all FPKM values. I've got a shopping cart here. I can select individuals of those, or I can click here and add all the files to my cart. So now that basically selected that whole population, and then I can effectively download this manifest, I can also explore those cases. So it means that I can get access to those. So that's now downloaded my manifest, and just see if I can pull this from my secondary screen. It's not letting me do that. But essentially that allows me to access those. And it will automate that process. I'm just trying to pull see the download is sort table image colors. I'll look at these cases in exploration. Right, yeah, so if I go to the repository, I need to reselect those. Right, so yeah, so I've added all of those files to the cart, and now if I go up here to cart, then I can select that I download everything from the cart. So if I click download cart, and now you can see it's come up with this this fact that it's going to take me some time to download it. Well, that's understandable. It's quite a big set of files. I might be able to cancel that. No, I can't. But I can also, what's important, as well as downloading these FPKM values, I also want to download the biospecimen data. So I can download that as a JSON file. I can download the clinical data, and I can download the metadata. I may well not be able to download. I'm not intending to download all of these, but I will stop it. But you can see now this gives me the opportunity to get into all of this data and consolidating this data, which is essentially molecular data for every individual in a breast cancer population. I think that's downloaded. If I want to look at one of these files individually, I'll do that. I'm going to download it. Now, let me see. Okay. And one of the file types is a tar file, but basically I can extract that and immediately see for that individual the sequence information. So what this is we have an ENSG code, and that's the ensemble genome. So that code effectively gives us a reference for each gene in the human genome. And so that's the first column. The second column is for that patient, that sample, what is the level of expression of that gene? So for example, here we can see that this gene has an expression of 7.23. Now, we can compile that for the whole population. So what that will give us is for a specified population, we will have the level of expression of every gene in the human genome. And from that, we can then build up a data file of that. So we can start to explore the relationship between particular genes and particular clinical groups, or particular transcripts and particular clinical groups in the population. So we might want to find the best subset of genes that, for example, correlates with survival or correlates with a particular subgroup of breast cancer, and so on and so forth. So this is the TCGA is the first one. It's very cancer specific, but it is a very big repository and very comprehensive and up-to-date data. So there's a big opportunity to analyze that. I'm going to I'm just going to cancel my downloads, because otherwise I will get about a terabyte worth of data if I'm not careful. But as you can see, that it's quite an in-depth approach, but it nevertheless is quite powerful in order to find information and get data for a cancer set of cases. So that's the first method. And the second method I'm going to show you is the Geo Data. So if I search for Geo Data, this is the gene expression Omnibus data set. This is also by NCBI. And I get this front page. So again, here it tells us all about getting started. It tells us about various tools that are available, how we can submit data here. So that's important. So for example, you can log in to submit data and submission guidelines. And you can see here, there's links to the Miami standards that are needed. The part I need to mainly look at is this bit, the keyword search. So, for example, I might want to look at data for tuberculosis. So if I click on search and I click on the number of results, this is every one of these 9,777 results is a separate study. Now, some of these can be absolutely massive because they look at every tissue that's ever been applied to on that array. And you can see here, I can search, for example, by number of samples. I can search by default order. And that's really just by the closeness of the hit. In other words, the more the record mentions tuberculosis or TB, then the higher ranked that will become. It's a bit like a Google a Google search. So I want to sort based upon the number of samples, high to low. And you can see here I've got various filtering tools that I can use. So for example, I've got here Homo sapiens, right? So if I click on that, that gives me 5,474 studies. And now I've just filtered to human tissues or samples. You can see here I can look at expression profiling by array, by methylation, and so on. So if I click on that, I want to look at array-based profiling. And I then also can customize in here for the tissue type, publication dates, and I also can, you should be able to search by array different array types, I think. Not at that point. That's changed. So what it does then is it brings up a page for every study which has a reference to tuberculosis in it, and we get effectively an abstract for that study. So I'll pick the top one. So we've got here the title: viral infection is a risk factor for TB disease in infants. And it follows the Miami standard, but so it's got a brief abstract. It's got the organism listed. It's got the type, expression profiling by array listed. It's got the sample number, and it's got the platform. And it's a text data file. So what it also has is a PubMed record as well. So because that's been published, I can get the paper that's behind that data. Now, it tells me things like the platform. So if I click on platform here, that gives me the GPL code. So that's a standard code for a particular platform. And as you can see here, the this platform is the Illumina expression bead chip. So this is a high density chip using the Illumina technology. It represents 47,000 probes. So it's relatively, it's not the latest, it's not the newest. It's been around since 2009, but it's still a reasonable array. What I want to do then is I want to look into this one a bit more. So I'm going to click on the link at the top, and that takes me into the Geo record for that study. So I can see that this data went public on the December the 5th, 2018. The species is Homo sapiens. I've got the summary, and I've got also a publication here. So if I click on the publication, I can now see that this is a Nature Communications paper. I've got the authors and all of the information I need, and I should be able to get a full text of that paper through PubMed Central. So I've also got a contact name and an email address. So this work was done by Adrian Hill's lab in Oxford. I can email that person. So if I have any questions about the data, then I can contact them. Now, what's important here is to see that this was last updated on January the 2nd. And that means that because it's relatively updated, I probably stand a good chance of being able to contact this person in order to speak to them. Right, so what I also have access to here are the platform details and individual samples. So if I click on a sample, I get the record for that sample, and I get the individual values for every Illumina probe. So these Illumina probes are, in effect, the identity of the gene that's expressed in this study. So again, I can get a reasonable record there. I can view the full table and so on. That is quite tricky though, because I might not give me the information I need properly. So if I want to get all of this data in one go, if I click on here, series matrix files, I'll click allow. It's now allowing me to collect. Sorry, this has gone onto my secondary screen. bear with me a second. So I'll just quickly share with my other screen. And so you can see it's allowed me here to open this, to get this file. I'm now trying to, it should be downloading. Yeah, and I'm now expanded that file. That's gone to my primary screen, so I'll have to reshare and go to that. Screen one. So you can see now from my primary screen, hopefully, I now have the series matrix file for all of those TB cases. Just going to create a bit of space. So this is the data. That's fine. Just so we can see it more clearly, I'm just going to drag and drop it into Excel. Right, so drag and drop that into Excel. Although it doesn't open. So this is now, I'm now opening this whole data series from a text file into Excel, and that allows me to show you the layout of it quite easily. It's taking a little bit of time here. Okay, that's got it. So these files tend to have a standard format. They have a header, which tells me all of the sample, or all of the study information, who's produced the data, the address, and things like that. If I delete that, that means I have then now a flat file which I can work with. I'm just going to stretch the width of those a bit, and now so it takes a bit of time because it's a big file. And now you can see we have a sample accession number, and that GSM number that's referred to here is the sample identifier. So that is a unique identifier for that individual sample. You can see this is a good study. They've given me a whole lot of things that I can look at. So, for example, I can understand the when the data was made public, the sample type, the tissue type, PBMC, it's human. I've got the subject identifier, which is from their record. I can see that some of these are unstimulated and some of them are stimulated with BCG. And we've got a case control, and we've got a CMV, positive negative status. We also have a vaccine that we've been given here. So we have also a treatment details, the extraction details, the array profile, and all of the contact information. So this is quite a comprehensive record. And at the top of the file, I have all this information, some of which might be interesting and useful for me to explore. So, for example, I might want to see for a set of for these cases, what were the differences between case and control. What I then also have are the ID references. So if I go right to the bottom, you can see we have 47,113 probes, so bits of the human genome, and those are measured for every individual in our population here. So every individual, we have a lot of GSM numbers. We have, in this case, 557 individuals for where that profiling has been conducted. So this is a pretty comprehensive data set. I could use this quite easily to study the differences in some of these subgroups, and I could understand, using some of the machine learning methods I use, what the comparison, what the key features might be. So I'm going to just a minute save this data, and just because I'm going to use this in a bit, and I'm going to save it as an Excel sheet, and I'll save it to my desktop. So in the next session, I'm just going to show you some methods that we can start to look at this sort of data to understand and get simple analyses to find the key genes of particular interest. So that's the Geo Data. I would say I'm going to show you how to use ArrayExpress next, but I would say because of the ability to download a single flat file, the Geo Data is much more user-friendly. We can quickly get data that's in a suitable format for us to do some quite detailed studies. With ArrayExpress, we have a different data set, but we have to assemble the data in some cases, and that can be a bit like a jigsaw. That can be quite challenging. So I'm just going to close some of these down and go to ArrayExpress. Okay, so for ArrayExpress, we just type ArrayExpress into the study, into Google, and we have again a welcome page and a splash page for ArrayExpress. What's quite interesting here is that it tells us how much data is stored on ArrayExpress. And this is currently 59 terabytes of archived data. So it's quite a big repository to start to work with. So you can see here that we have various tools. Again, we have projects, links, and the like. We can browse data. So we can look at all the studies that are stored on there. You can see there are 73,000 different studies. As with the Geo, there are guidelines and help to submit data. And again, using things like the Miami standard. There are help pages to help people who haven't encountered before to search for this data, and there were online tutorials here. Programmatic access is quite interesting and useful. So, for example, if you want to develop a Python program that scrapes data from ArrayExpress, then this will tell you the steps you need to do. So you can build up an API effectively to automate the collection of data from ArrayExpress. And then there's help on submitting data. So there's always a news page at the beginning, so you can see here ArrayExpress is moving to biostudies, and so that's a major point, and so this will become biostudies in a while. It will just be redirected from ArrayExpress. So the tools and techniques will probably remain the same. It's just a different branding almost. So if here, for example, I want to search for lung cancer, you can see here that I can, it's quite a detailed search. I can look for lung carcinoma, lung lymphoma, lung sarcoma. So you can see that I can it gives me a kind of a dynamic search. And I can search for lung cancer cell or lung cancer cell line. And so you can see that there's quite a lot of options even in the search. But I'm going to look just for lung cancer now. So if I click on search, there we go. So once I've done my search on lung cancer, then I come up with a whole load of records here. And you can see that I've got 1,352 experiments here. And I can then filter the results here pretty much the same as I filtered before. So if I click here, I can click on filter. So I can, in here, I can choose the organism. So I'm going to choose Homo sapiens. I can filter based upon experiment type. So I can look at amplicons, DNA, metabolomic, proteomic, and RNA. I'm just going to select RNA. And here I can select on the assays that are used. So it could be an array, could be sequencing, and there are more mass spectrometry or cell methodologies as well. So fluidigm-STEAM1 multifluidics is effectively a way of looking at individual cells, and we're getting more and more single cell sequencing data being put on this sort of platform. So I'm going to select sequencing assay for now, because that will take me to something else as well, which I want to demonstrate. And you can see here I've got the opportunity to check if I want an array, the array type. So it breaks it down by Affymetrix arrays. You can see massive numbers of different types. And I can also look at Illumina arrays and so on. So there's quite a lot of arrays here. The common ones are at the top. So you can see things like HU133 plus 2, human 133 plus 2, HD133 plus 2, exon 1, gene 1, are the common ones that we would use here. But because I'm using a sequence assay, that's kind of irrelevant. So if I click on filter now, you can see that the sort of studies I've got here are RNA-seq of coding RNA. It's highlighted the organism and it's highlighted the assays. So I want to find the richer data sets because I want to have more cases. So if I click on assays, that now sorts the data out for me from a high number of replicates to a low number of replicates. So in this study, we have the accession of the study. And so the European studies now tend to be called MTAB studies. You can see we've still got some of the GEOD ones as well, and these tend to be sort of the older studies, because the EBI and GEOD do no longer align. So it's two separate repositories now, really. So the latest Geo studies won't appear on ArrayExpress. So, okay, so I want to look at RNA-seq, and let's find something suitable. It's a cell line study. I don't really want a cell line study. I want ideally a tissue study. These this one is a single cell study, so I'm looking not at that at this point. Okay, so we have here RNA-seq of primary tumors from 79 small cell lung cancer patients and 7 normal tissue. So if I look at that record, it tells me that there is RNA data and there's atlas data. So this particular one links me to another repository, which is the European Nucleotide Archive, ENA. So if I click on this, I get the same sort of form as before. So I've got again species, the sample identifiers, the protocols, the description, the experiment type, and then I've got the study. So I've got the PubMed paper for this, and so I can get access to that. And what I have here is a slightly different format. So I have a file which I can download, which is the investigation description. So if I click on that, that gives me effectively what would be the header of the GEOD file. And I've got the full description here. What I also have is what's called an SDRF file, the sample data relationship file. So if I click on that, then this is a table which gives me the clinical information of the study. It's not so easy to see there. I'm going to download it. Into downloads. And if I now drop that into Excel, it's not liking that. I'll open it from Excel. No, open. Yep. So if I now open this, you can see that I have, for every individual in the population, I have a record. I can see, for example, their source name. So that's the unique page sample identifier. I can see that tissue's lung. I can see their gender. I can see their age. I can see their ethnicity. I can see whether they're smokers. I can see whether they have small cell lung carcinoma, tumor profiling has been done, and so I've got all of this information about the population. Let's close that one. And now I have additional data. I'm not sure what that contains, but also I have a link here to the expression atlas, which shows me, allows me to filter out particular genes that are up and down regulated from the study. But also I have access to ENA, and this gives me effectively another link and allows me to download the FASTQ files, raw data, for each individual in this population. Does it give me? Yeah, so I can also check to see if I have any. Yeah, so these are all, if you like, FASTQ files which I can download via FTP. So that gives me access to the raw data, and this is another repository which is from the European Nucleotide Archive. Now, what's quite nice about having the FASTQ files, if I really want to get into that in depth, is that I can realign that to the genome, and as the genome becomes updated, so I can keep realigning, and it also means that I can get much more depth of information rather than the pre-processed files. So I can dig down into those files and look at the results at the gene level. I can look at the individual transcript levels, so the parts of the genes, and I can look at individual nucleotides for those as well. So I can really dig down deeply, if I want to, into these studies. That takes a lot of time, though, and a lot of data processing. So that's one record, which is from ArrayExpress. I'm just going to filter using a different filter to show what happens when we use an array assay. And I'm going to look at one particular array, which is the 133+2. And now if I filter, I have anything mentioning lung cancer and where we've profiled with an array and where we have assays. So if I now look here, I've got a number of studies. I'll just pick one. So again, I have the same layout as before. The array, it lists the array. So I can click on that, and this file here, I'll go back to the primary record. The file here gives me the ADF, the array design file. So what that tells me is for every probe on the array, and in Affymetrix they use this underscore AS or underscore ASAT, underscore AT, it gives me the identity of the probe, and it will give me the identity of the gene, effectively. So that's one thing I can download here. I have, as before, the investigation description, so I can download that. I have, as before, the sample data relationship file. And I can now download individual raw files. So these are the cell files. I'll look at one of those. Pull that up. It's downloading, so it takes a little time. And I can also download a processed set of files as well. So these are the ones that have been through the normalization process. Let me see. Just taking a little bit of time to download these. So while they're downloading, does anybody have any particular questions that they want to ask me at this stage? After this bit, I'll go on to another methodology. So this is the raw files. You can see each one of these is a .cel file. These are the unnormalized files, but what that means is I can then go back in, and with technologies like GeneSpring, I can then renormalize those, or I can co-normalize to different data sets as well. So I can start manipulating the normalization to merge files, for example. I've also got, I'll just close that one down. I think it's that one, and then the cell files. So if I click on processed, I'm hoping that's going to download reasonably quickly. Yeah. So here I have effectively, for each individual in the population, a separate file which gives me the ID reference, so that's the gene name, and then the amount of expression of that. So I would have to go back, and this is with the difference between Geo and between ArrayExpress. I would have to go in and then merge all these files together. So that is all well and good, but the other issue we face here is we have these codes, and they don't really tell us much about what those genes are. So we can do various things there. One thing we can do is we can get the array library file. So if I go Affymetrix U133 plus 2, you can see it's come up here with U133 plus 2 annotation file, and I can get that through various sources. I tend to use Thermo Fisher because I can normally download, here we are, the support file for that. So click show more. I want the yeah, so I want the library file, human genome 133 plus 2. Right, so I can get a free registration for this, so I've forgotten my password. Okay, so I just had to reset my password, but if you you can set up an account quite easily. Sign in. Yeah. So. So what I've got now is, hopefully it'll keep me in the same place and not have to go back. So let's cancel that. Right, so sorry about this. Right, so now if I go here, right, so now that should be downloading, yes, the library file. So what that gives me is a way to convert from the Affymetrix codes for that particular platform into the gene names. So that's important because otherwise, without that, this becomes kind of meaningless. The other way to do this is if I have a small number of genes, copy those, and I want to find out what they are, then I can go to another page, which is gene annotate home. And basically that allows me through the gene cards, which is an information source, to search for a particular gene set. So if I paste that in, and I can search by probe set, I can submit that query. I need to look at +2, sorry. So I can submit that query, and you can see what it's done is found these gene codes and the gene names for the things I put in. So gene cards is also a useful tool because now, for example, let's say I want to find out what calcium binding protein 4 does. If I click on that, I can now get the full gene cards record, and that tells me all about that gene. It tells me what it does. It tells me where it's located. It tells me the chromosome it's located on. It gives me a list of products which I can find for antibodies to the protein target. It tells me the cellular localization. It tells me the string code. It's a huge amount of information here. I regularly get questions from people who are not biologists who say, well, I don't understand enough biology to understand what the genes do. And to be honest, I think probably nobody really understands enough information to know what the genes do. But it's just through things like gene cards where we can start to get an insight into that sort of thing. So by finding a set of genes with a computational method, we can then go and explore what they're doing further. So hopefully my library file will have downloaded. Yep. So if I click on full HTML2 library files, Thought they put them in text before, but so I'm just going to open it in a text page, see what we get. Text edit. No. Maybe let's have a look at the file. For some reason, this has changed. It used to be just a text file. So I'll search it. I'm just going to search again. So HQ133 is 2.0. Right. Right. These files. Right, so just try library files. It's trying to get me to install software, so it's not the what I want. I can try another source, but it's not so easy that way. I'm afraid I have to look into that. So the key point here is this gives us effectively plenty of data to work with. We can find identities from gnan.home as well, and so this gives us data to work with. So now just quickly going back to the data we have here, I'll show some quick methods to find out what the key features are, what the key genes are. So let's say, to begin with, hypothetically, I have a case and a control for TB. So I'm just going to copy those and paste those out. So in my simplest possible experiment, I have a case of TB and a control of TB. I'm just going to get rid of all of this for now because I don't need all of that. But this is our control and this is our case. What I want to do is find out what the differences are between expression levels of the genes for case and for control. So the way I'm going to do this is I can do one method when there's a pair like this, which is to regress the values in the case against the control. So case on the y-axis, control on the x-axis. So first, just to illustrate that, I can plot an XY scatter plot of the case values and the control values. And you can see that there's a very dense region of expression, but then we get some things, for example, this one, which are up and down regulated in in the case compared to the control. So that's one way of doing it. But I want to be a bit more quantitative than that. So one of the things I can do is I can do a regression of case and control. Now, there are various ways of doing that. If I Excel, then one of the add-ins I can add into Excel is a data analysis tool. If I click on regression, click OK, then I can set that regression up to regress the case example against the control example. So these are my controls. So I want those to go on the x-axis, so I'll put case on the y-axis. And I'll select, oops, I'll select case on the x-axis. So what I've done is set up the x and y range. I'm just going to click residuals and standard residuals, because that will tell me how far every gene is away from the consensus, if you like. So if something's highly overexpressed, then it will have a positive standard residual in case, and if it's underexpressed in case, it'll have a very low standard residual. So if I click OK on that, it does the regression. And you can see here, I have effectively the standard residuals for that population of points. And they range between -12 and +12. So what the standard residual here represents is the number of standard deviations that a particular gene is expressed over or under the parity line between the two states. So, you know, something here, for example, has 12 standard deviations in case overexpression, and other things have 12 standard deviations in case underexpression. If I just copy those, then I can actually put those next to the points. Shift them down. And now let's get rid of those two. What I can do is I can sort based upon that standard residual. So you can see here now in the TB, with respect to TB case, this particular gene is downregulated 12.59 standard deviations. If I go to the other end of the spectrum, then this particular gene here is upregulated to 12.84 standard deviations. So I might want to know what that is. So if I click copy, I can even just drop that into Google. To find out what it is. I want the ones that are not at. Okay, so that looks like a ribosomal RNA 28S protein. Just check that with. So if I look at the other, that looked like to be a ribosomal protein. If I copy this one and search for this one, so this is a hemoglobin subunit alpha, HbA1, HbA2, also known as. So I can quickly look at some of these identities, and I can see what key genes are coming out. Obviously, I'm just looking at a couple, and there's a lot more to look at. If I go back to Geod, I might be able to find the array design file. So if I put in GEOD GSE 98550 98550. Yeah, so I've now pulled that record back up by just putting that accession number in, and I'm just going to look at here. So I've got the platform. Just quickly click on that, and hopefully samples. So if I click here, I can download the library file from here. So if I now drop that into Excel, for some reasons I can't do that. So if I open that from my downloads, so now I can, from my downloads, open that file. Yeah. So this is my now my library file. So if I search for this, so Edit, find. So we click Find Next. Now, so I've got my library file from Geo, and now maximize that. I've got the Illumina code. I've got the sequence, and you can, yeah. So I can see here hemoglobin alpha subunit 2. I should have. And I think I need to load it and import it as in a slightly different way. So if I click Open, Downloads, go to that, Open. I need to set it as delimited, not add. Next, finish. Yeah, so that's better now. So from that I can get the gene name, I can get the sequence, and I can align my library with all of that if I choose to do so. So that is another reason why Geo was better. It's got more up-to-date library files than ArrayExpress. So that gives me another way, by using regression, at looking at multiple pairwise comparisons of the data. Another way to do it is I can look at, for example, a t-test. So let me see, and just pull a few of these up for now. Shall I tell you what I'll do? So first thing I'm going to do is just get rid of some information that is not useful in the first instance. Delete. I just want a case versus control. Delete. So I'm going to sort based upon row 3. So if I sort, custom sort, right, left to right, okay, row 3, okay. So what that's now doing is sorting based upon row 3, so I should have all my cases together and all my controls together. Either thinking about it or not done it. Try again. Yep, so if we color now all of the cases, so if we color all of the cases, let's color those red, and we color all of our controls blue. Then we can run a t-test to look for things that are associated with, in this case, tuberculosis. Obviously working on quite a lot of cases here and controls. So. Right. Sorry, it's taking my computer a little while to deal with all of this, but hopefully. There we go.
I think there's too much data properties.
Yeah. Anyway, I think that's got it. So if I run that and color those blue, then we're getting there slowly, sorted. Yeah, good. Right, so if I want to do a t-test in Excel quite quickly between these, if I say equals t.test, then what I can do is I select the array. And then I select the next array, which is there. And then I want to set it as, I'm going to set it as a two-tailed test, and I'm going to set it as unequal variance. That has the least level of assumptions. Yeah. So that gives me my p-value here. So I can, if I double click on that little box there, that will give me a p-value for every single gene on the whole array. What I want to do is now sort based upon that p-value. And I want to sort based upon the I think it's UL. So that's working its way through there. So the idea is we're effectively sorting on the best genes that have a distribution that has minimum overlap between TB and non-TB in this case. There is a challenge with that because we're looking at nearly 50,000 probes, and there's always a chance that by random chance they might be false discoveries. So what we have to do then is we have to correct for what is called multiple testing. So we can use a method called Bonferroni Correction. And what that does is it, well, in its purest form, it adjusts the threshold for significance. And effectively, we can adjust the thresholds for significance by dividing that, the p-value, let's say it's normally 0.05, by the number of tests we're doing. So obviously, if we're doing 50,000, 0.05 divided by 50,000 is around about 10 to the minus 6. So that is quite a low threshold for significance. The other way we can do it, of course, is multiply the p-value we get from our test by the number of tests we do as well. And hopefully that's just done. Still thinking about it. The one thing I would say as well is that working with such big data does take time. And when we start looking, well, the next series, the next couple of lectures I give will be on machine learning. When we started out in this space with machine learning, some of our algorithms were taking 6 months to run to get a result. So it can be quite time consuming if
So even with the very strong GPUs, it will take so long.
Well, we're much better now, so we can screen with our GPUs now. We can screen 40 million molecular combinations an hour. So the technology has come on a long way since that start. So I don't know if I wanted to try and see if I could get a distribution of these p-values, but it's taking a little while now. It may be I have to leave it running and then show in the next session, but
yeah, that's also a possibility. If it takes time, we can.
So if I can just summarize what we've covered then. So we've covered the basic principles behind repositories. We've gone through the Tcga interface to show how we can access and download data from Tcga. We've gone through the Geo data set and shown how we can access and download data from Geo. And we've gone through the ArrayExpress portal to show how we can access and data from ArrayExpress. I've also shown how we can get library files. There have been some issues with those being up to date on ArrayExpress, but also just touched a little bit on some of the basic statistical methods we can use to explore these data sets to find information from them. In the next lectures I'll be talking about machine learning and AI and how we can apply some machine learning and AI methods to this sort of data. But I'd be happy to answer any questions as well.
Oh, great, thank you. I think it was a quite long session, and you showed everything live, which is great. You'll be happy to know that Abhishek is also in this group right now. Abhishek Sen Gupta, probably he has got some training with you already.
Yes.
Abhishek, anything you want to comment? Abhishek or any other participant, if you have any query for Professor Graham.
It's quite interesting. The website SAR searched can be shared with us. We will go to that website and do it relaxedly to know better about this. We have quickly run through that. Anyhow, he has referred a larger number of websites. That website can be listed and shared with the participant team, and that's my request, sir. Thank you.
What I would say is we will have this video recorded, I guess, and follow through the processes if you get a chance, and if you have any questions, then I can answer them in the next session.
Right, so I will actually upload these videos also shortly and make it available to all the participants so that they can get some more time to go through the contents. And of course, then they have a week time to browse through the information, and then if they have more specific queries, we can take in the next week.
I think my sorting is just finished, actually.
It is, yeah.
Oh no, maybe not. I tried to move it.
This one done now? Okay.
I'll wait till next week and I can show you the distribution of those things. I tried to move the file and then oh no, maybe yep. Okay. Yeah. So
okay.
Can you see my screen?
Yeah, it is visible.
So you can see here I've got a set of p-values which range from 8.49 times 10 to the minus 10 right the way through to 0.9999. So that's everything from significant to virtually identical. The interesting thing here is that we should really be just taking the things that are different at a p-value of 10 to the minus 5 or less because of false discovery. So you can see really we've only got 21 differential genes coming up here through that big population, and so we can see which ones those are in the library file. So the point being here is we've got a rank order of genes. I'm not sure how consistent this data is because of this relatively low number of significant, features. But it might be the methodology. We could, if we use machine learning, we might find some more there as well. So.
Great, wonderful. So thank you so much, Professor Graham, for giving this very informative session. And I'm sure many of our participants will be now able to go through the more repositories, more resources, and try to then use that data for their own analysis.
Excellent. Yeah, as I say, if you have any questions, please mention them in the next session or contact Professor Srivastava and I'm sure he'll pass them on to me.
Right. Professor Graham is again coming back next week. So then if there are additional queries, we can also take that time. So, all right, that's pretty much for this session. So thank you, Graham, once again. And we look forward to have your AI machine learning session, which will be further to this particular today's session in the next week or next